Hello dear viewer, welcome to my YouTube channel. In this video lecture we will discussing about Clostridium bacteria. There are four medically important species of Clostridium bacteria, that is Clostridium perfringens, Clostridium tetani, Clostridium botulinum and Clostridium difficile. But in this video lecture we will specifically discuss Clostridium perfringens bacteria. Well, the Clostridium perfringens bacteria causes two distinct diseases, that is gas gangrene and food poisoning. Gas gangrene, also known as Clostridial myonecrosis or simply myonecrosis. Gas gangrene is a bacterial infection that produces gas in the tissue in gangrene. This deadly form of gangrene is usually caused by Clostridium perfringens bacteria. It is medical emergency. There are about 1000 cases of gas gangrenes occurs in the United States annually. Now gangrene or gangrenous necrosis is a type of necrosis that caused by critically insufficient blood supply, or it can cause by localized death and decomposition of body tissues resulting from obstructed circulation or bacterial infection, or in other words we can say tissue death due to loss of adequate blood supply. Sometimes bacteria invade such tissue and accelerate its decay. It is characterized by foul-smelling discharge, surface and subsurface discoloration, usually in lower extremities. The causative agent of gas gangrene is an anaerobic gram-positive spore-forming bacilli bacteria of the genus Clostridium. Clostridium perfringens is the most common etiological cause of gas gangrene. Here are some other species of Clostridial that cause gas gangrene. They include Clostridium bifermentis, Clostridium septicum, Clostridium sporogenes, Clostridium novae, Clostridium felix, Clostridium histolyticum and Clostridium tertium. Transmission of Clostridium perfringens. The Clostridium perfringens spores are located in the soil. Their vegetative cells are the member of normal flora of colon and vagina. Gas gangrene are associated with war wounds, automobile and motorcycle accidents and septic abortion. Now, pathogenesis of Clostridium perfringens. The organism grow on traumatized tissue, especially muscles, and produce a variety of toxins. The most important toxin is alpha toxin or lecithinase toxin, which damages the cell membrane including those of erythrocytes, resulting hemolysis. They have degradative enzyme which produce gas in tissues. There are at least 20 enterotoxins of Clostridium perfringens which are, well, here are the most important entero or exotoxin and their biological effects. Number one is alpha toxin, which are lethal, lecithinase, necrotizing, hemolytic and cardiotoxic. Beta toxin, they are lethal, necrotizing. Epsilon toxin, they are lethal and permease. The permeases are membrane protein, a class of multipass transmembrane protein that facilitate the diffusion of a specific molecule in or out of the cell by passive transport. 4 is iota toxin. It is lethal and necrotizing. Number 5 is delta toxin. It also lethal and hemolysine. 6 is phe toxin, which is hemolysine and cytolysine. 7 is kappa toxin, lethal, collagenase, gelatinase and necrotizing. 8 is lambda toxin, which are protease. 9 is mu toxin, that is hyaluronidase. 10 is nu toxin, it is lethal deoxyribonuclease, hemolytic, necrotizing. Their lethal effect is tested by injection in mice. The clinical finding of Clostridium perfringens include pain, edema, and cellulitis that occurs in wound area. The crepitation or a crackling or rattling sound indicates the presence of gas in tissues. Hemolysis and jaundice are common. Shock and death can ensure. The mortality rate are high. Treatment of Clostridium perfringens. The drug of choice is penicillin G. The wound should be debrided to remove dead, contaminated, or adherent tissues or any other foreign materials. In prevention, the wound should be cleaned and debrided. Also, penicillin may be given for prophylaxis. The second disease caused by Clostridium perfringens is food poisoning. Clostridium perfringens are one of the most common cause of foodborne illness or food poisoning. CDC stated that these bacteria cause about 1 million foodborne illnesses in the United States annually. The Clostridium perfringens are present in raw meats, poultry, dehydrated soups, raw vegetables, and spices. In the United Kingdom and in the United States, Clostridium perfringens bacteria are the third most common cause of foodborne disease. Now about the transmission and how they contaminate the food items. The spores of Clostridium perfringens are located in soil and contaminate food. The Clostridium perfringens bacteria heat-resistant spores survive in cooking and germinate, and also the organism grow its large number in reheated food, especially in meat dishes. Pathogenesis of Clostridium perfringens. The enterotoxin causes marked hypersecretion in jejunum and ileum in small intestine. A heat labile proteins are produced by some strain of Clostridium perfringens. When greater than 10 raised to power 8 cells in contaminated meat are ingested and sporulate in small intestine, the enterotoxin is produced. The enterotoxin of Clostridium perfringens are identical to a protein in the spore coat. Now, clinical findings. The food poisoning disease caused by Clostridium perfringens has an 8 to 16 hours incubation period and is characterized by watery diarrhea with cramps and a little vomiting, but the disease resolves in 24 hours. The laboratory finding is not usually done and there is no assay for toxin, but large number of organisms can be isolated from uneaten food. In treatment of Clostridium perfringens, we provide symptomatic treatment to a patient. For example, if someone have vomiting, we must give them anti-vomiting drug, or if someone is dehydrated because of watery diarrhea, we must rehydrate with electrolytes, that is sodium potassium chloride. In treatment, no antimicrobial drugs are administered because it resolves in 24 hours. There is no specific preventive measurement or safety measurement. The best preventive measurement is that the food items should be adequately cooked to kill the organism. Thank you, dear viewers.